Ambassador program. We still have some people chiming in, but we will start with our overview of the program. The Water Ambassador program is a partnership program between Martin County, the University of Florida IFAS Extension, and the Florida Sea Grant program. It is a stormwater education program designed to teach residents of Florida, the Treasure Coast, as well as visitors of how to do their part to protect water quality in the state by limiting their pollution associated with stormwater. If you happen to live in Martin County and you see something that doesn't look right, you can report stormwater issues or any illicit pollution discharges through this QR code or through the phone number there. Believe it or not, we are nearing the end of our 2026 program. We have, following today, we have three more presentations. One in August, hosted by the Indian River Lagoon National Estuary Program on the My Plastic Diet. And then we have two in September, talking about issues and challenges with communicating about water in the state, and then also the Lagoon Drift Citizen Science Project. As always, we'll be recording this presentation and posting it on our YouTube page. We will put the link in the chat. And we ask that you turn off your audio and we will save the questions in the chat bar. So all questions will be saved to the end after the presentation and we'll get to them in the order in which they were received. And with that, I am excited to present our speakers today. We have a joint presentation talking about upside down jellyfish as a bioindicator of pollution in the IRL. Our two presenters are Marie Strader and Natalia Lopez. Marie Strader is based out of Texas A&M University, and her lab studies how ecological and evolutionary processes play out in response to human-induced changes in the environment. The research in our lab aims to establish links between genotype and phenotype that will aid in predicting ecological and evolutionary trajectories of marine invertebrate taxa. Natalia Lopez is a 2025-2026 Gulf Science Policy Fellow with the National Academies of Sciences, Engineering, and Medicine. During her term, she will be hosted by GCUS and serve as the Gulf Coast Acidification Network Coordinator. Natalia brings more than a decade of experience in marine ecology, oceanography, teaching, and mentoring. She earned her PhD from marine science in the University of South Florida, where her dissertation focused on the ecology of upside-down jellyfish and their potential as bioindicators in Jobos Bay National Estuary Research Reserve. So I want to thank you so much both for willing to present to the water ambassadors today. And I'm going to stop sharing and turn it over to you.
All right. Can everybody see the screen all right?
Yep, looks great.
Okay. Thank you, Lisa and Vincent, for inviting us. And we're really excited to talk about a topic that both Natalia and I are very passionate about, which is upside down jellyfish, which hopefully by the end of this webinar, you'll have learned quite a bit more than you knew before about them and what their relevance is to local ecology. Okay. Okay. Just to follow up really quickly on introductions, and then we also kind of wanted to let everybody know what the connection between the two of us is. I'm an assistant professor at Texas A&M University. My journey to get there went through education at a variety of different states, including Oregon and University of Texas at Austin and California, and I'm now at Texas A&M. And my research lab there, we focus a lot on understanding range edge dynamics of upside down jellyfish, as well as other organism environment interactions in marine invertebrate species. So we work with a variety of different species in the lab. And Dr. Lopez?
Hello. Can everybody hear me okay? Okay. So, yes, as Dr. Strader said, my name is Natalia Lopez Figueroa. Newly acquired PhD, fresh off the boat, probably almost a year ago. I did my bachelor's in the University of Puerto Rico at Humacao in coastal marine biology. I have a master's degree from Hampton University in biology, where I did zooplankton taxonomy. Uh, and now I'm doing and then I did a PhD in marine science with the upside down jellyfish as potential bioindicators. And if I had to say any focus research, I don't have a real focus in the sense of like, I started with stable isotopes and how can we use them to trace like rainwater and different water sources in a estuary. I've done genetics work with DNA. I've done taxonomy, which is how to identify little organisms in to the lowest, like what are they and what did they do. But in the basics, I'm an ecologist, in interactions between organisms and their surroundings. And so I've done pretty much a lot of different things. So but I'm an ecologist through and through.
Yeah, and we don't necessarily work together, but we met at the International Cassiopea Workshop. So this is a workshop that our research community puts on every year. It's in Key Largo, Florida, and it brings together researchers that work on this upside down jellyfish as a system to study a variety of different things. So this jellyfish is a unique system. There's mathematicians that are using it to study fluid dynamics. There's evolutionary biologists that are using them to study the evolution of sleep. They've been used as a model to study symbiosis in the context of coral bleaching and a variety of other things. So it's a really small meeting where we get together and we just share a bunch of ideas and our research around this model system. And this is a picture of all of us from the workshop a couple years ago. And so, because of that, we've all kind of been able to come together and find connections between interests and research that have led to different collaborations. So, okay. So, just to start off with, we wanted to get an idea of a few questions from the audience. So if people could sign on to this real quick, you can use the QR code, you can use the web link there, and we have two questions, and then we'll show everybody the responses in a minute. Natalia, you're muted.
Of course. 6 years in doing webinars and this happens. But as you can see, we're looking at our live poll of have you ever seen an upside down jellyfish? We have a lot of people that have seen them. That's really cool. There's around 40 something participants. So for terms of time, Marie, you tell me. Seems like
Okay.
most of our participants know what an
Okay,
upside down jelly is. So we have
that's
a
great.
baseline.
Okay, let's go to the next one.
Okay, so our next question is, have you ever seen an upside down jellies in the Indian River Lagoon? Ooh.
Ooh.
Thanks for everyone for engaging. This is awesome. So for as of now, the polls are saying no, Marie.
Yeah, but there's a lot of people that have. And so
Yes,
maybe I'll take it.
I'm very excited to see these results.
Yeah, me too.
As a true scientist, we're excited to see results. Yes.
Okay, I'll go back to sharing the PowerPoint. Okay. Oh, of course. Okay. Okay.
Alrighty. So what are jellies? But I see this crowd is very well versed in that, so I'm just going to skim through this. So we're going to use the term jellyfish interchangeably. We know that they're not fish, but jellies are essentially gelatinous free-swimming organisms within the phylum Cnidaria. So that's just like how we group them in what we call the animal kingdom. And these particular animals are very known for having stinging cells, and I know I can't see you, but raise your hand if you've ever been personally victimized by a jelly in the beach when you're trying to enjoy yourself. Um, and so but they use these as to defend themselves, and this is something a very characteristic aspect of them, right? They're found worldwide and they play a huge ecological role in the ocean, not only in the food webs, but also in the dynamics and the ecology of the ocean in general. So they're not just pests. It's like mosquitoes. Like, why do they exist? Well, we all have a purpose. So we often see them as simple organisms, it's just a blob that swims. However, they have very complex life cycles and adaptations. Like, for instance, box jellyfish have really complex eye structures that they can see color and distinct light. So, and they're very avid swimmers. So they might seem very simple, but they're very, in my opinion, they're pretty incredible to do very much a lot of different things without having a brain, for instance. And they can form large swarms called blooms. Marie, can you? So because we know that they form blooms, but it's very kind of not as much. We don't really know as much about their blooms, specifically near the coastline. They're essential contributors to a process called the biological pump. So essentially, the transport of carbon from the surface ocean to the deep ocean, and they thrive in changing environments as well. And we think of jellies as these robust organisms that can withstand a lot of change. So we associate, oh, there's a bloom of jellies, that means the water's bad. Not necessarily. They respond to different anthropogenic activities, such as increased nutrients or decreasing pH. And their blooming conditions and their nature remains unclear. Even though we know that they bloom, there's still a lot that we don't understand. And for that, we need in order to understand blooms, we need to know their life cycle, because sometimes you just see no jellies, and overnight you just see that they form these big swarms. So a big enigma, if you will, is that we need to learn more about their bloom dynamics.
Okay, so we're going to focus this talk on specifically the upside down jellyfish. So this comet is commonly called the upside down jellyfish, but the taxonomic name for biologists is Cassiopea. And this is the only known jellyfish that we would call benthic. So that means it likes to sit on the bottom. So it sits on the substrate in the ocean. And you can see that video here at the bottom of the upside down jellyfish where it's sitting on the bottom. So at first glance, it might not even look like a jellyfish, because people would expect a jellyfish to look, say, like the video up at the top, the typical jellyfish. But an upside down jellyfish is a true jellyfish. It just sits on the bottom. It can move about a little bit. It can still swim. If it gets knocked up, if it gets disturbed, it can pulse up and it can move around a little bit. And there's a lot of things interesting about this jellyfish, but it's becoming invasive in a lot of parts of the world. So there are regions around the world in the tropics where they never found this genus of jellyfish before, but all of a sudden it appears, and then all of a sudden it becomes a nuisance. Um, and this is partly because their mode of dispersal is not completely known. I'll talk a little bit about the life cycle in the next slide and talk about what we think might be happening with creating these blooms. And this particular genus of jellyfish is found globally across the entire world in the tropics. So this map down here is showing all of the instances where Cassiopea have been observed. And the green observations in this map are showing citizen science observations. So observations of people just snorkeling, diving on the coast, by their boat docks, so on and so forth. And all of the Blue squares are incidences where scientists have reported or studied Cassiopea. So, like Natalia said, how can this jelly bloom form? So, if you see a bloom of jellyfish, if you have just a ton of these giant jellyfish that seem to come out of nowhere, how could that possibly happen? So, one way is by thinking the answer is probably within its complex life cycle. So, we see the big jellyfish, but the earlier life history stages are actually quite small and almost microscopic. So, let's see, do I have a pointer here? Pointer, laser pointer. Okay, so if we have the big jellyfish here that has its tentacles, it has a lot of different colored appendages. It produces eggs and sperm, like a lot of other animals. These eggs are very, very small. The females will keep them attached to them until they develop into swimming larvae. These swimming larvae are so small, it's like a speck of sand in the water. You can't find them in the wild. They're so, so small. What happens is these larvae, they swim about, they can swim in the plankton, but eventually they will detect some kind of cue in the environment and metamorphose into a polyp form. So this polyp form looks a little bit more like an anemone, right? And you can see the picture over here. And this picture under the microscope, all of these little dots here, these fuzzy little dots, are small, recently settled polyps. So this is with a microscope image, and they look clear, they look beige, so they're practically impossible to see in the wild. And the other thing is that these polyps and these planula, they tend to want to settle on things like decaying mangrove leaves. They can settle on boats, they can settle on turtles, they can settle on other kinds of leaves, all sorts of things. Some things that can move, like boats. So if they settle on a bunch of boats, that could be a way that these animals are transported pretty much undetected to new environments. And these polyps, they can bud asexually, so they can reproduce very, very fast through budding. And they don't need a sexual life cycle in order to do this. So they can produce large amounts of polyps in a very short amount of time. They also are symbiotic with a symbiont that triggers them to go through another phase of their life cycle where they do what's called strobilation. So what strobilation is where this polyp basically transforms itself from a polyp form to a disc. So you can kind of see the disc starting to form here in this picture that becomes the jellyfish part. So the part that looks like a jellyfish. So it develops this disc, it pops off the disc, so now it's free swimming. The early stage is called an ephyra, and then it develops into a large adult like a medusa. So, like I said, if you see a bunch of these large jellyfish and they came out of seemingly nowhere, it doesn't necessarily mean that they all swam there. In fact, they probably didn't all swim there. They probably were part of this strobilation cycle. So there was probably a huge mat of polyps somewhere in the environment. Something in the environment triggered them to go through this strobilation and produce a ton of baby jellyfish that grew into large jellyfish. So a big mystery about these blooms is where are these polyps? How are these polyps getting around? And what initiates these polyps to create this stage of the life cycle that we would actually see and interact with?
I want to add
So,
something, Mary.
yeah.
Sorry. What also makes this very interesting is that these jellies in particular, they have a 101 ratio in the sense of like it's one, like for each polyp, there's one medusa. A lot of like moon jellies is like the part of the strobulation looks like a lot of little umbrellas stacked on top of each other. So one polyp can give a lot of little jellies. It's even more impressive that they form these blooms that Marie is saying because they what they seen in the lab is that per one polyp gives away one medusa. So imagine how many polyps have the like could be in there. Take it away.
Yeah, it's pretty impressive. And so in Florida, Cassiopeia have historically been commonly found in the Florida Keys, as anybody that's been to the Keys has probably found them. And they live in a variety of different types of habitats. So they're also commonly known as the mangrove jellyfish because they like to live in mangroves. You can see a mangrove jellyfish here along the mangrove roots. But they also live on sandy bottoms. They live among seagrasses and other types of macroalgae. You can see these pictures here. It might look initially like it's just a bunch of grass or algae, but there's actually hundreds of jellyfish here in this picture. Similarly, on the bottom here, you can see one here. There's hundreds of jellyfish in this picture. And then they can also form close associations next to high high human densities. So this is a jellyfish here that's close to a boat dock. It's really hard to get pictures of them near boat docks and boat channels because of the turbidity of the water. But anybody that's been around boat docks and boat channels in the Florida Keys knows that there's often Cassiopea there. So a few other things to note is that these Cassiopea, they have stinging cells, and these stinging cells are what helps them eat. So it allows them to catch prey, to catch things like small fishes, other invertebrates, things like that. But what happens is when they're disturbed, they can release their mucus, and within that mucus are a bunch of stinging cells. So this is what would be called a contactless sting. So you don't have to touch the jellyfish to get stung by them. You can just They can just be jostled, release the stinging mucus that can just float amongst the surface. So it's possible for you to just be swimming and you don't even know there's jellyfish there, but you could be stung by their mucus. And this also can be a detriment because these Cassiopeia can occur at really, really high densities. And this can impact tourism on beaches. It can impact the ability to get into the water to do any sort of recreational activity. And it can just be a general nuisance. Um, Natalia, do you want to talk about the second half of the slide?
Yes, of course. So on the right side, you see a lot of jellies. So the first picture, which is on the left, is Cassiopea at Hobo's Bay. So the water looks in Hobo's Bay like the picture that Marie showed in the near the boat dock. It looks like it's turbid, but when you dive in it, it's pretty clear. And unless you are going to do like an exploratory, like kind of snorkel first, if you head out of the boat and step on the benthic, so on the ground of or the seafloor, you might be stung. And as Marie said, not necessarily you need to touch them to get stung. And we've all experienced that if you swim near upside down jellies, you know that you are going to end up all itchy. And some people develop really severe allergies to them. But the same way that they form these blooms, they can also massively die off. Like on the picture on the right side, this is in Fajardo, Puerto Rico. Both of these pictures were taken near mangrove channels, like mangrove lagoons. And this jelly is often called the mangrove jelly. And they can play an important role in their ecosystem as ecosystem engineers. So they achieve this status by, significantly affecting local hydrodynamics, nutrient transport, and zooplankton communities. So the little microorganisms that are in the water column. And their ability to mix water and transport nutrients from the seabed makes them a key player in shaping these shallow coastal environments. Because as you may imagine, when these all die off, they're going to release whatever is in them. And most of that is carbon, right? And all of the other nutrients that they have, but it also all that gelatinous material. So what happens after a bloom, it's still also something that we're trying to figure out. So their pulsing behavior does enhance the mixing. And even though that they do these pulsations with the bell, they lift up nutrients from like the kind of like recycled nutrients that are already used up and kind of settled in the sediment. Um, and so these supplies food to help sustain the food web as well. So that's why we're kind of pitching in this concept that they might be considered ecosystem engineers because of the way that they shape the ecosystem.
Okay, so this kind of begs the question, where are they? How can we track where they are? Are they coming and going? How can we kind of monitor this? So there's a lot of challenges with monitoring range shifts and also where species are, species distributions, and how they change through time in a lot of marine organisms. So there's fishery surveys that are done by institutions like NOAA and others. These fishery surveys usually encompass large taxa, and they don't include everything. It's just too comprehensive to include in a bunch of surveys. And also, surveys are only a snapshot in time, so they don't necessarily give us an idea of how populations and species distributions are changing through time. So there's not a lot of information that exists. And so one of the things that we've turned to is thinking about, well, what are people seeing, like actual people in the community seeing? And one of the ways that we can track this is through citizen science. And so we used observations of Cassiopea that were documented in iNaturalist. So iNaturalist is a website where anybody can just report sightings of different animals that they see. And they can submit pictures, and they submit pictures that are dated and they have a location associated with them. And so we can use this as data basically to determine where have certain species been found, where have others not been found, and how can we look at this through time, since this is a database that people have been submitting observations to for about a decade now. So Cassiopeia is a good species for this because they're found close to human establishments. They are coastal, like extremely coastal. They're associated with boats and things like that. And people tend to interact with them more than they would a jellyfish that lives in the middle of the ocean. Nobody's really going to interact with that very often. So what we did in our lab was we collected 12,000 observations of Cassiopeia that were observed through iNaturalist in Florida from the years 2000 to 2024. And for each one of those observations, we characterized the phenotype of the animal, so what it looks like basically, what color it is, what size it is. We also looked at the habitat type and we georeferenced that with satellite imagery. And we also collected temperature data from NOAA temperature loggers. And so what we wanted to know is anecdotally, we'd been hearing that Cassiopeia had been found more farther north than they had been previously. So we wanted to see if this was actually something that was substantial that was actually happening. So over here on the side are some of the observations that have been found. So these are observations. The first one is south of St. Pete. We have some near Fort Pierce and then through Miami. So if you go on, this is a really rich data set for us scientists to be able to use to answer a lot of the questions that we have about where are they found, what are their dynamics, where are they coming and going, where are established populations that are there for a long time. And so we've also started, we really are encouraging people to report sightings of Cassiopeia, especially in the Indian River Lagoon area and some of these more the middle of Florida sites on both sides of the coast. So if you do see them, I highly encourage you to report your photos on iNaturalist. The photos don't have to be great. It's actually very hard to get pictures of these guys that look good because they're squishy and blobby and weird. Underwater pictures are great if possible, but even just mentions of them or some kind of any kind of evidence is good. So the scientists really rely on people that are out there in the field that are interacting with these coastal ecosystems to give us some information that we can use to answer important scientific questions. So what did we find? So this is a GIF here of Florida, and so we looked from the year 2000 to 2025, and historically, and they were only found in the Keys, but through this period of time, we found that they slowly started to find observations farther and farther north every single year. So prior to 2015, there were no observations of Cassiopeia north of the 26th latitude. But since 2015, there have been observations further north every single year to the point where there's now year-round established populations in both Tampa and then Fort Pierce on the Atlantic coast. And so when we did this study, this was in 2025, and we were like, okay, these are established populations. Every time we go out there, we can find them. People were very reliably reporting seeing observations at these sites up farther north. So these are what we're going to call our range expanding sites. So they were never there before. They're now there. So what's driving this? We think a big thing that's driving this is temperature. So these are tropical species. They're tropical species, so they're actually very well adapted to heat. They can handle a hot summer. They can handle a hot summer day. They do have thermal limits. They do melt when it gets 40 Celsius. So they do have a heat limit. But we were really interested in the fact that they're probably more sensitive to cold. And so what we did was we took a NOAA temperature loggers across a lot of sites where we found that were adjacent to where we had been finding jellyfish. And as everybody in Florida knows, there was a very cold January in 2025. And so this is some of the temperature data from that event from January, early January through the end of January. And so these different colors represents loggers that are in these different quadrats of the state. So we have northwest, northeast, southeast, and southwest. And so we counted how many cold days there were, what the minimum temperatures were across these different regions. And so what we considered a cold day was anything that was below a mean temperature of 15 Celsius. So 15 Celsius is what we already know from the jellyfish as being kind of a sensitive spot. So in the Northwest, for example, there were 22 cold days. So these are days that we presume the jellyfish won't be able to survive. In the Northeast, there were far fewer cold days, and it didn't get as cold. And in the Southeast and the Southwest, there was it definitely got cold, but it didn't get as cold, and there was just fewer cold days. So what we did after this was we surveyed 30 sites looking for Cassiopea. So we went to a lot of these sites that had been year-round populations. People had very reliably found them, and all of a sudden they were gone. They were gone all over the board. So all of these different X's are sites that we looked for jellyfish, and the X's represent where they were absent. So this population that had established in the Tampa Bay area completely wiped out. And because of the temperature data, we suspect that probably the polyps were wiped out too. On the East Coast, on the Atlantic coast, we found one medusa during this time period. And based on the temperature data, we suspect that the polyps probably survived. So if the medusa were wiped out, the large jellyfish-looking ones, it's possible the polyps were still retained. Um, and the populations in the Florida Keys were perfectly fine. We found really high densities like usual. So one thing we're doing in our lab is we're looking at the genetics of these populations, and we're monitoring through community science and citizen science data to kind of see when they come back. And so these Cassiopea populations are fluctuating in Florida, but also everywhere across the globe. So this is that map I mentioned before. The green are areas where there have been citizen science observations. The blue is where scientists have observed and reported them. And there are a few places around the world where they've become invasive and they become quite a nuisance. So one example is in Hawaii. So this is a fish pond that local communities use. They've designed these kinds of fish ponds to be able to collect fish in high densities for food as a food source. For whatever reason, Cassiopea got in there and bloomed. There was massive blooms of Cassiopea in these fish ponds, and it totally wiped out the fish populations. So this is just an article from the local news. Their conservation effort to actually remove all of these jellyfish, they called it jellyfishing, where they scoop up and try to get rid of as many as possible. But now that everybody here knows about their life cycle, you can collect all the medusa, but that doesn't mean that you're getting rid of the polyp populations. And the polyp populations are practically invisible, and they can induce a bloom pretty much whenever the conditions are right. Similarly, in Brazil, in the southern hemisphere, there's also incidences of jellyfish blooms of Cassiopea, and invasions in parts of these coastal ecosystems that they were never found before. And so another interesting component of this is they tend to be clonal lineages. So it's one individual that's producing a lot of clones. So it is kind of like Attack of the Clones with these jellyfish. We don't know why. So that's another thing that us as scientists are trying to study. Um, okay, so at this point, I think you want to change over the remote control, Natalia. Okay.
Alrighty.
Okay.
So I conducted a literature review, which is a fancy word to saying that I read a bunch of papers, dissertations, books, and I surfed the web for different sources of like what type of publications are out there, like what are we publishing on these jellies. And interesting enough, this map resembles one part of Marie's map, which is where have we had studies of Cassiopeia done. So on the top right side, you see that there's like different colors. So if it's pink, or kind of reddish is less than 5 studies. And if it's kind of like dark purple or more of a darker color, it's more than 20. So as you can see, it's very widespread where we have conducted these studies. But if we amplify that, we can see that Florida and the Caribbean has been basically leading the field of Cassiopea research per se. But there's still a lot that we don't know. And as Marie mentioned, these jellies are expanding. So they're undergoing to what we call latitudinal expansion, right? And so based on that, we need to have more studies about them, because for what I compiled, which is nearly 200 studies, 71% of those were performed in lab-based environments. So in a lab-based environment, you can control everything, right? But when it comes to the field, it's kind of it's a little more trivial because you don't know exactly, right? Like how are things going to behave or if they're going to be there. So oh wait, I have to ask you to unmute. There you go.
No, this is good. This is good. So, just to kind of transition here, these Cassiopeia are common everywhere. We know that populations can boom and bust, but then the question is, can we monitor these populations and can their presence or absence tell us something about the health of our ecosystems and something about water quality? So this is something that Dr. Lopez studied for her dissertation, which is why I invited her to give this webinar with me.
All righty. Let's see if this allows me to move because I'm having kind of a hard time with it.
Okay.
All righty. So when we hear the word bioindicator, what does it actually mean? So there's a lot of different definitions for what a bioindicator is. But in essence, what it is it's an organism that exhibits a change in their appearance or like their structure or function linked to a biological effect, whether it's a contaminant or any physical/chemical changes in the water. And the literature has many definitions to it, and it's encompassed in basically essentially this is what it is. But what do we know about Cassiopea as a bioindicator? Does it have the capacity to be a bioindicator? Well, checking the literature, Cassiopea has responded negatively to herbicides. So when exposed to different herbicides, specifically those that target like the photosystem, which is like the part of the photosynthesis aspect, they respond negatively to it. They've also responded positively in the Bahamas to nutrient input. And I think the author of that particular study is among us today, Dr. Betsy Stoner. Dr. Betsy Stoner's group also looked at them as a microplastic sink, which is a great tie-in to remember that webinar that's going to come up in the water ambassadors. So because they're filter feeding, everything that goes in the water column is going to go through their filtering system, right? And they have found that microplastics can be bioaccumulated in their guts. They've also responded negatively to heavy metals in the water, as well as they follow human activity. But what does following human activity really mean? Well, in Brazil, they showed that after they dredged a pond, they came up that was next to a mangrove system. They dredged the channel towards that pond, and then they saw a bloom happening basically overnight. And the same thing happened in Hobos Bay, which is what drove me to Hobos Bay to conduct a study, because around the world we've been seeing these type of behavior. But that was part of my dissertation, and today I'm just going to focus on what I did for the main project, which is answering the question, are upside down jellies bioindicators of nutrient enrichment in Hobos Bay, Puerto Rico? This was a project funded by the Noah Margaret Davidson Fellowship, and it was a collaboration between the University of South Florida, University of Puerto Rico, and Hobos Bay. So Hobos Bay is located in the south of the island of Puerto Rico. It is a high human activity area because the coastal zone in Puerto Rico is it's defined as probably 100 to 200 meters from the line of the high tide. However, Puerto Rico has a lot of people and there's a very big, dense population. This also used to be a sugar mill in the lands of where Hobos Bay is at. So a lot of the people that worked in the sugar mill established their families in there. So these are also inherited properties. So because of all that human activity that it's been subjected to over history, it became a very interesting site to look at because we do find Cassiopea in Hobos Bay, and managers and fishermen told me that they have been seeing an increase in them for the past maybe 2 to 3 years. And so that led me to study Cassiopea in Hobos Bay. Now, how did I define human impact? There's different ways that we can do this, but I based mine human impact index based on Dr. Betsy Stoner's 2011 study. And it's like in a 2 square kilometer radius, I counted the buildings that are within that radius. And if it surpassed 100 or 200, I deemed that high human impact. If it was like in between that, less than 100, it would be moderate, and if it was less than maybe 80 to like more so like 50 to 0, it would be low human impact. So based on this is a map of the reserve. So the high human impact is on red, and it's located in these areas of the reserve. So on the right side where you see those 2 points, those 2 sites are near river outflows, and those rivers or those creeks, sorry, they flow through this urban area. So these are all urbanized areas that are constructed from people that have lived there and not necessarily had permits for it. Same thing happened on the left side when you see the entrance of the channel of Las Mareas, and you see a news article tied to there. Those are illegal constructions that were new, and this became a very controversial / interesting part of this study because the managers wanted to know if the Cassiopea were indicating something about the people that were doing these illegal constructions, right? They didn't have septic tanks. They didn't really have any plumbing system that went through a treatment plant. So you can imagine all the other things that might have brought. So these areas, because of the buildings around them, were deemed high human impact. The yellow part is moderate because even though there are some constructions, these actually are remains of the agricultural sites of when the sugar mill was active. And some of these agricultural lands are still active. And then we have the low human impact, which means that it's not that there's no human impact. It's deemed low because of how the estuary flows. So because of how the estuary water flushes out of this area, it is not as stagnant or close to the land processes like when we have river runoff. It's not as close. So because of that, we deem this as low human impact. So the question for the study was, can we use Cassiopea as bioindicators of nutrient enrichment? But before we know this, we need to know, is there nutrient enrichment in Hobos Bay? First off. Second, if so, if there is nutrient enrichment, when does it occur? And are Cassiopea responding to these nutrient pulses? Note that I'm not putting in any, is it responded positively? Is it responded negatively? So these are the main questions that we wanted to answer. And in order to do that, as great ecologists that we are, we have to look at everything around it. So very simple, right? Not really. So we looked at the water quality assessment, which is very important because these jellies are surrounded by water. So my group did a study where we looked at we took temperature, salinity, dissolved oxygen, and pH. We also looked at nutrients in 2 different aspects. We looked at nutrients in the water, where we looked at nitrate, nitrite, and phosphate. But we also collected macrophytes. So macrophytes are algae or seagrass, basically aquatic plants that you can see with your naked eye. In this case, we collected mostly algae and seagrasses that were on site. But why did we collect these? Well, water moves, and water is a snapshot of how the nutrients were there at the exact time that we were sampling. However, marine macrophytes like seagrass and algae accumulate nutrients over time and gives us like a longer version of how the nutrients look when we're not there, when we're not sampling. So it gives us a better idea of how the nutrients are usually when we're not around and in other times that we're not sampling. And then I thought it was a good idea, and it is, but it's very complicated, to do random quadrat tosses and take pictures to estimate the population density, or what we call percent coverage. We used a quadrat. I'm not sure if anyone's familiar with it, but we built a quadrat. Basically, it's a big square made of PVC tubes, and we had little lines that were 10 centimeters apart. And I tossed this 10 times around the area. And I took a picture of each quadrant to the best of my abilities, adjusting for the lens and the lens distance between the bottom and where I was taking the picture from very carefully, because if you disturb these jellies, you're going to get stung. And I looked at not only the area coverage of the jellies, but we looked also at the fauna and the flora that were around it. So if we saw any sea urchins, or if we saw any fish swimming around them, and what type of seagrasses or algae were present. And we did this randomly across the site. We made sure, however, that we were not sampling during the same, we didn't oversample or overestimate in the same area by kind of swimming maybe like 6 to 10 feet away from where we were sampling. And sometimes we would mark these as well, so we didn't have to cross samples. And these sites were pretty big. And then after that, I would take these pictures and put them in a free website that's called Beagle. And Beagle essentially calibrates the images so you can estimate the area, but you can also measure the jellies, which is what I did as well, because I also wanted to see the size distribution of these jellies. So the photos were annotated individually. That took forever, but it was very interesting to look at them and see how big they are or how small they could be. We have tried to avoid overlapping the polygons, because the polygons is all the lines that we were forming. We call them polygons. And we calibrated with a laser point feature using each quadrat. That's why the little lines came in handy. And we estimated the size using the ruler function with those laser pointer features. Now, this was kind of tricky, because when they form blooms, they can be stacked on top of each other. So again, we can only estimate how many jellies were there and what was the area coverage. We didn't really count when there were blooms, like on the bottom right. We didn't really count that how many jellies were overlapped on top of each other, because it was pretty difficult to figure out. So in a nutshell, this is a very brief results slide, but if you have more questions, I am always happy to talk results with you guys. But essentially, what did the nutrients say? So for the nutrients, the more impacted sites, which were the ones on the top right, near those riverine inputs and that illegal constructions, they showed that they had higher dissolved nitrogen and phosphorus. Aquatic plants, so the macrophytes, told us that the nutrient levels varied widely across sites. We did find more nutrient concentrations in the moderate impact sites, which was interesting. But at the same time, they were accumulated over time. So there is some runoff, but because there's mangroves there, the mangroves are doing their job at filtering the excess. The seasonality. So in Puerto Rico, we have 2 seasons, wet season and more wet season. So some of the sites showed a strong seasonal change, especially in phosphorus. And phosphorus is used a lot in agricultural land. And that, as I mentioned, this estuary per se has a lot of creeks that flow in it. That's how it feeds the fresh water. And so the seasonal changes were more driven. It's like we saw more phosphorus when there was riverine runoff, which makes sense, right? And so for the water conditions, the temperature and salinity differed across sites, with lower salinity at the higher impact sites that were adjacent to creeks. That makes sense because fresh water is flowing in, and a wider salinity range in the Las Mareas community where the illegal constructions were at. Now, we don't have an explanation per se for this, but what we told the managers in our final report was that maybe they need to look at different aquifers or groundwater inputs that might be influencing this, or maybe there's a lot of freshwater input from another area they haven't really identified. So this kept us this was a very interesting thing. Now, this looks very busy, but I will guide you through it. We will become experts in reading this type of graphs, right? I'm going to do my best to explain it so we can
Natalia,
all understand it.
we,
Yes.
this is, we have 10 minutes left.
Okay. All right. So this is called a principal component analysis. And essentially it's telling me how similar or different are my samples and sites. So the months are in different shapes and the colors tell us different sites. The principal component 1 tells me about, oh, I lost you guys. Are we still there?
We're
Okay.
here.
Okay. It's trying to show the relationship of my samples to each other. And what you can see, the principal component 1 tells me what is mainly driving my variability. So it's 31% of the variability is explained in this axis. The principal component 2 explains 21%. So if you put these together, if you sum that up, these percentages, it makes that 50% of the variability of my samples is explained in this graph. Now, when the points are close together, it means that if these samples and sites are similar to each other, so I can tell about these sites and months that the dissolved oxygen and temperature might have been similar. But when the points are really far apart, it's telling me that these months and sites were very different from each other. So what does this mean? The bell size positively correlates with nutrients. So just to say it in plain language, just because of turns of time, when there's more nutrients, the Cassiopea gets bigger. Essentially, we found bigger bell size that the nutrients in the percent nutrients and the nutrients in the water showed us that they were positively correlating to the growth of the jellies. Now, what did it say about the percent coverage or the density? We did not find any environmental drivers that were positively correlated to Cassiopea coverage. On the other hand, we did see opposite correlation with different nutrients from the bell sites. So in plain language, as you can see, the percent cover is on the other side. So they're like completely opposite from each other, the arrows. And essentially, when we had more salinity, plus we had more nutrients, we saw the Cassiopea population or the percent coverage decrease. So going back to the bioindicator aspect of it, did Cassiopea provide insight on both positive and negative ecological impacts? No, because we were not measuring whether it was positive or negative. Did it display a measurable response? Yes. Did it reflect ecological responses to abiotic changes? Yes. Did it demonstrate resilience and maintain fitness under both short and long-term environmental changes? Yes. Did it detect environmental changes and contamination impact? Yes, to environmental change. We did see changes seasonally and with the nutrients, but Hobos Bay, because it has so much runoff, nutrients are not, it might not be the main driver of what is driving those populations increase and decrease. So we just looked at the environmental aspect, specifically nutrient enrichment. So in a nutshell, nutrient enrichment follows the seasonal variability, specifically the wet season. The percent coverage varies very much differently from the bell sites. Hobos Bay shows very variable hydrological components, and it explains complicating efforts to establish a long-term bioindicator. Oh, And despite the ecologic knowledge that we lack, Cassiopea does hold promise as bioindicators for Hobos Bay. So the question remains, are they responding to environmental variability? Yes. Can they serve as bioindicator in Hobos Bay? Potentially. This was the first study of in Hobos Bay, and it's probably the seventh of its kind. So we still need to establish a bioindicator. We really need to focus more on producing more studies.
Maybe I really want to answer some
Yeah.
questions. So just to kind of summarize, basically we would like to do a similar study in the Indian River Lagoon, and there's lots of similarities and differences between these different ecosystems. And the question as to can they be a bioindicator in the Indian River Lagoon is still unknown. But Dr. Lopez has done a great study that gives us some hints about what could be happening in other ecosystems, but would need to be followed up due to the different uniquenesses of each type of environment. Um, yeah, let's see. Should we should we open it for questions? Because I really would love to.
Yes.
Um,
Great. Thank you both so much for a really fascinating presentation. For those of participants, you'll note that I launched our quick evaluation poll. We do ask that you take 3 seconds of your time to just complete that for us. And onto the questions. The first one goes back to sort of I guess, the conversation that you were having about water temperature. And Mark asks, water temperature rarely in this area rarely goes, as low as 14 or as high as 40. So could it possibly be air temperature and not water temperature?
Oh, that's a good question. So, probably not air temperature because, but then again, like wind patterns can affect the temperature of the water in really, really coastal ecosystems. And you're right, it doesn't really go to those extremes very often. And that's why the Cassiopeia are perfectly fine there most of the time. But what is affecting them are these extremes. And so what's happening as global climates shift is the extremes are becoming more they're occurring more often. Like in 2025, there was a huge cold spell in Florida that was really uncharacteristic. And then this happened again this last winter, where it also dipped really, really cold into Florida. So, yeah, normally that's why they're usually typically fine, but it's some of these extremes that are happening more than usual that can knock them out. Yeah, that's a good question.
Great, thank you. So currently, seagrass is recovering in some regions in the IRL, Indian River Lagoon. If we see large blooms of Cassiopeia in these recovering areas, should we be concerned for the seagrass?
That's a good question. You want to get to that one?
So it's interesting because when I started working on this, I almost did this study in Florida Bay. And essentially, what the Fish and Wildlife Commission said was that there was sort of a succession. Whenever they saw seagrass dying, they saw Cassiopeia kind of moving in. But then as Cassiopeia moved in and died, they saw like these cyanobacterial mats. So there's sort of a succession there. So that's very interesting. And maybe they're telling a different story in the Indian River Lagoon. So who knows? You have to do a close you have to see closely like what happens after the Cassiopeia leave. That would be my next assumption.
But the Cassiopeia can shade the seagrass, so they can negatively impact the seagrass because they both rely on photosynthesis. So if the seagrass is covered with Cassiopeia, it won't be able to photosynthesize as well. So there are a lot of interesting interactions between those two organisms for sure.
So should you be concerned for the seagrasses? Maybe.
Maybe.
Yes.
Okay, thank you. We are at 1 o'clock. So, with respect to all of your times, I want to say thank you to the participants and to Marie and Natalia. If you want to stay on to answer some of the other questions, we'd be more than happy to have you. But I do want to acknowledge the time and just say Thank you, and thank you all for joining us again. And we hope that you will come to our August webinar on my plastic diet. Okay, so the next question, this is just a clarification of the results. So smaller numbers of jellyfish, but bigger, fatter ones in response to nutrients, correct? Is that a
Correct.
good interpretation? Okay.
Yeah, so they are bigger, but they're less.
Okay. And then someone is saying our county, so Martin County, is discussing allowing chickens in our seaside town. Do you believe that chickens, and I guess the waste associated with them, could cause some of these blooms?
That is a really interesting question. Based on the studies that have been done, I would say it's definitely possible. I think there's a lot more complexity to it. It's going to introduce particular nutrients, but then how those nutrients are recycled and moved around in the system and the hydrodynamics could make an impact. But it might increase nutrient, it might lead to new localized nutrient enrichment. I would expect that it would, which um, could promote the jellyfish to increase in population size. But what do you think, Natalia?
I believe that it depends also on the density of the chickens as well and the usage of it. I think I don't know if this is all parts of Florida, but Florida does regulate the fertilizers as it is. So I think that if there's a regulation for, like when what to use when, it'll depend on the density of how many people like are having these chickens and what it takes to have their care.
Thank you. And
Lisa, there's one
yeah,
more question there's a question before that from
did I
Elise.
skip one? I'm sorry.
Yes, there's one.
Oh, okay. Elise asks, has there been a study in the IRL indicating pollution culprits and its effects of seagrass?
That's a good question. I don't know. Um, I'm unsure.
Okay. I will say that there has been some studies about pollution in the Indian River Lagoon and the sources of those pollution. At least maybe we will revisit that as a topic for next year, since it's been a while, I think, since we've covered it. So
Mm.
thanks for giving us a note for our next series. Um, Katie, so in response to the results of jelly larger jellyfish, Katie asks, do you think that that's because the larger jellies are outcompeting the smaller ones for other resources?
That could be a potential answer. I think it's very complicated to point one because doing statistical analysis on interactions is pretty difficult, or at least that's what I've encountered. But it's a possibility. It might be also fitness, like larger. It's maybe beneficial for reproductive purposes to be a larger individual. So it might be that they're taking up more space, right? They're gathering the area, the nutrients, so they might be out competing them, but it might also be something that might not be controlled in the site per se. Maybe the availability of seafloor because they need a bare sediment to attach because of their suction cup, right? So sometimes they will try to set on seagrass, but they can't really like do that suction cup and stabilize in the seagrass on top of the seagrass per se. So it could be a possibility. It could be that maybe there's something in the water that affects more the smaller individuals rather than the larger individuals, bioaccumulation wise. So there's a lot of different factors that could affect it, but that's a potential one.
Great, thank you. And then in response to the chicken manure question, one of our participants, I guess, is interested in sort of joining that conversation on Facebook and asked if they could use your response. My response to that is to reach out directly to Maria and Natalia via email to get, if they want to provide a quote, that it is maybe a bit more thought out than than what was just presented here, if you both are okay with that.
Yeah, yeah. But I mean, the real question is we don't know, and it depends, which is, of course, not really a helpful answer. But yeah,
Okay,
we'd
great.
be happy
Thank
to
you.
discuss further.
And I think that's it for the questions, unless Vincent, did I miss another one?
No, I think that's really good. The only thing I want to sort of comment on is related to the chickens because there is a proposal for having backyard chickens in our area, specifically Martin County. It would have to be a really specific situation with regards to, a waterfront property where that could be the
Mm-hmm.
case. And it's sort of would be sort of, I would think, be, kind of lumped in with all the other potential sources of nutrients that could potentially get into our waterways and cause blooms of any sort, whether it's
Yeah.
jellyfish or algae. So that's, I would definitely hesitate in saying, that, as a generality, that backyard chickens are going to lead to these, massive jellyfish.
Yeah,
So
I agree. It's all going to be relative. Like it
mhm.
might be a small blip compared to other nutrient inputs, right?
Lawns.
Yeah, yeah, exactly.
Yeah.
It's all kind of gonna be relative. And you're right, it could have no effect on the Cassiopeia, but it could cause other types of blooms, like other
Mm-hmm.
types of organisms, bacteria, protists that could bloom in response to high nutrients that it might affect more than the jellyfish. So yeah.
Yeah. Yeah, I'd be cautious about using that as any kind of argument, you know,
Yeah.
against having backyard chickens.
And if it's just Stuart with three chickens, that's probably okay.
Yeah.
I'm sharing. A friend of mine was a former postdoctoral fellow in Harbor Branch, and he did study like harmful algal blooms in the Indian River Lagoon. So he might be able to answer some things and that you may have about maybe not contamination per se, but maybe nutrient inputs or other sources that might be affecting the Indian River Lagoon as well. He did this study a while ago, like back in 2020, 2021, when we were all locked up, but he was outside. But yeah, like I think that that study may be able to answer some questions that are around it. But the main thing that I'm proposing and that Marie and are talking about with our field studies is that we want to encourage more people looking at these animals, reporting them in iNaturalist if they can. So that helps us a lot because we're not there every day. And a lot of the livelihoods of the people in Florida and other coastal areas are depends on their on the ocean on the coastal side. So it's a very big help, and it will allow us to go a long way. So observations, the origin of species from Darwin was based on observations, right? So observations are crucial for us to understand after a rain event, I saw this, and that you're a scientist. That's exactly what we try to figure out. So I think that's what we want to stress in order to better understand this as a tool for managers and other coastal like water quality monitoring. It is very dependent on having eyes in the field.
And just to confirm, because we did get a question in the chat, so it's not an actual link, it is just the app, the um, phone app iNaturalist, where you receive all your data. Okay, so
Yeah.
okay, great, thank you. Well, thank you again, and thank you all for joining us. This was fascinating topic, and hopefully you'll get some more Cassiopeia pings in the IRL and Lake
Yeah.
Worth Lagoon.
Yeah. Thank you so much.
Thank
Thank
you,
you.
everybody. We appreciate it.
Thank you. Thanks, Marie. Thanks, Natalia.
Thank you.
Thanks.
So